Welcome back to Parasitology. Hi, I'm Christopher, and in this video, we are moving into Enteric Coccidia and Cryptosporidium. This is where small stages can create very large clinical problems. The main organisms for this video are Eimeria, Cystoisospora, Cryptosporidium, and Cyclospora as public health context. The practical rule is simple. Match the host, match the stage, match the syndrome, and then ask if there's a public health angle. Small oocysts, big outcomes. Before we go further, let's clarify vocabulary. Cyst, oocyst, and egg are not the same thing. A cyst is usually a protozoan survival or infective stage. The classic example is Giardia. So if you hear cyst, think protozoal package. An oocyst is a coccidian or apicomplexan protozoan stage. In this video, that means Eimeria, Cystoisospora, Cryptosporidium, and Cyclospora. These are not worm eggs. They are protozoan stages with their own life cycle logic. An egg usually points you towards helminths like nematodes, cestodes, and trematodes. So remember, cyst equals protozoan, oocyst equals coccidian protozoan, and egg equals worm. Please do not call protozoan stages eggs by default. Somewhere out there, a parasitologist feels a deep disturbance in the force every time that happens. Now let's introduce the apicomplexan triangle. The word apicomplexa is useful. Api or apical points to the apex, the tip, and complex refers to a group of specialized structures. These organisms use the apical complex to invade host cells. So the name itself reminds us these are cell-invading protozoa. The triangle gives everybody a simple map of a life cycle that can otherwise feel like a blender full of vocabulary. There are three big processes to track. First, schizogony or merogony. Schizo means to divide, mero means part or segment. This is the asexual amplification side of the triangle. This is where one parasite becomes many. In other words, this is the parasite's regrettably effective copy machine. Second, gametogony. This is the sexual stage, where the male and female gametes are formed and fusion leads towards a zygote. Third, sporogony. This is where the oocyst becomes an infective package, often in the environment for Enteric Coccidia. Use this triangle as a sorting tool. Apicomplexan life cycles can appear complex, but placing them into the triangle reduces confusion. As is common, we start with a taxonomic parasitic chart. Eimeria, Cystoisospora, Cryptosporidium, and Cyclospora are coccidians, which live within the clade alveolata. Remember, clade is a useful set of organisms that have a common ancestor. Sort of like 23andMe, but for parasites. Hey, they need their family too, don't judge. Now we start with Eimeria. A small etymology note. Eimeria is named for the German zoologist Theodor Eimer. Sadly, this is not one of the names that tells you the life cycle. Sometimes etymology gives you a helpful clue, and sometimes it gives you a dead German scientist. Both are part of the journey. Clinically, Eimeria is one of our classic Enteric Coccidia. It is usually host-specific, and it is very important in production animals and poultry. Use the triangle here. Oocysts are shed. Under appropriate environmental conditions, they sporulate and become infective. The host ingests sporulated oocysts, sporozoites are released, and the parasite goes through rounds of asexual amplification and sexual development in intestinal cells. The practical diagnostic clue is fecal oocysts, but the clinical interpretation is bigger than that. You need the host species, age, syndrome, parasite burden, and sometimes lesion location. The oocyst is not a full diagnosis all by itself. It is the beginning of the interpretation. Whenever dealing with an apicomplexan, use the triangle. Here we can identify that two sides of the triangle occur within one animal, the ruminant. The third side of sporogony develops within the environment. The location for schizogony and gametogony can vary by Eimeria species and host species. Two Eimeria species generate visible gross pathology through megaschizont formation. Eimeria gilruthi generates visible changes in the sheep abomasum, and Eimeria bovis in the cow small intestine. Clinically, think young animals, diarrhea, dehydration, poor growth, and in more severe cases, dysentery or tenesmus. Fecal oocysts support the diagnosis, but always interpret them with the animal's age, signs, and timing. Eimeria species are usually specific to host species, so match the host before guessing the species. Age matters as well. Calves, lambs, and kids have their own clinically important patterns. Different species localize to different intestinal sites, and that affects the lesion pattern and severity. That is why site is not trivia. Site explains signs. The clinical impact can range from subclinical production all the way to diarrhea, dehydration, poor growth, and severe or hemorrhagic disease. Young animals and heavy burdens are the big risk pattern. For diagnosis, fecal oocysts support the diagnosis, but numbers alone can be misleading. Interpret oocysts with host, age, clinical signs, management, and parasite burden. Poultry Eimeria can get complicated quickly. Eimeria acervulina is classically upper intestine and is one of the more common species. Eimeria maxima is more mid-intestinal. Eimeria necatrix is associated with major lesions in the proximal to mid-small intestine, while the sexual phase is completed in the ceca. Eimeria brunetti extends into the more distal intestinal regions, including the lower small intestine and rectal or cloacal areas. Eimeria tenella is the classic cecal species. Do not overread this slide as a perfect GPS map. Biology rarely respects PowerPoint boundaries. But as a clinical framework, site is very useful. Cystoisospora is the dog and cat coccidia pattern, especially in puppies and kittens. You may see the older terminology like isospora in some settings, but don't get thrown off by historical naming. Unsporulated oocysts are shed in feces. In the environment, they sporulate and become infective. While fecal oral is common, paratenic hosts can contribute to infection. When an animal such as a rodent encounters this oocyst, the parasite insists in somatic tissues, hence the name Cystoisospora. Clinically, think young age, diarrhea, crowding, stress, and variable severity from subclinical to enteritis. Diagnostically, fecal flotation can detect coccidian oocysts. Now we get to Cryptosporidium. The etymology is useful. Crypto means hidden, and sporidium points to a spore-like stage. So Cryptosporidium is basically the hidden little spore, which is a polite name for something that can cause a lot of diarrhea and public health trouble. The oocysts are tiny, and they're immediately infective when shed. Water and environment matter. This parasite is resistant to chlorine and is a leading cause of waterborne disease in the US and associated with recreational water activities. Transmission is fecal-oral, and exposure can occur through contaminated water, feed, hands, fomites, or surfaces. Clinically, think enteric diarrhea, especially in neonates and immunologically vulnerable hosts. Calves are especially important in veterinary medicine, and public health relevance is common. Now we separate the Cryptosporidium species pattern. Cryptosporidium parvum is the classic veterinary high-yield species for neonatal calves. The big clue is calf diarrhea plus zoonosis or public health concern. Think contaminated water, contaminated environment, calf outbreaks, and people at risk after exposure. Cryptosporidium canis and Cryptosporidium felis are the dog and cat association. Same genus, different host clues. Cyclospora is mainly a parasite of humans, but as a parasite, veterinarians are often asked about it. Think public health, watery diarrhea, travel-associated or outbreak-associated disease, and contaminated food or water. Cyclospora oocysts are shed unsporulated and need time in the environment to sporulate to become infective. That separates it from Cryptosporidium, where oocysts are immediately infective when shed. Let's review. Production animal or poultry plus host-specific coccidiosis equals Eimeria. Anatomic location helps identify the species. Young dog or cat plus coccidian oocysts plus diarrhea equals Cystoisospora. And don't forget its paratenic potential. Calf plus diarrhea plus zoonotic concern equals Cryptosporidium parvum. Dog or cat sporidium, think Cryptosporidium canis or felis. Human watery diarrhea outbreaks through food and water contamination, think Cyclospora. Well, that wraps up this video on Enteric Coccidia and Cryptosporidium. Thanks for watching. I'm Christopher Lee, and what you know today protects everyone tomorrow.